Hello friend, welcome to Science for Sleep. Tonight, we're going to spend some quiet time together exploring amber. Most people have heard of this crystal-like substance, but not many know that it is a slow geological process that began in living forests and continued quietly through deep time. There's nothing you need to follow closely, nothing to remember, just gentle explanations unfolding at a steady pace. Now, let's begin. Amber is often described as a gemstone. It's polished, worn, collected, and admired for its warmth and color. But amber is not a mineral. It does not crystallize under pressure deep within the earth. And it is not stone in the way granite or marble are stone. Amber begins as something much softer. Amber begins as tree resin. Resin is a sticky substance produced by certain trees. It flows when bark is damaged, seeping slowly from cracks, cuts, or broken branches. In a living forest, resin serves a purpose. It seals wounds. It protects exposed wood from moisture, fungi, and insects. It acts as a quiet defense system, flowing only when needed. When resin first emerges, it is aromatic and fluid. It can drip, pool, or coat the surface of bark. Sometimes it hardens quickly in open air. Other times it remains sticky for days, catching dust, plant fragments, or small organisms that pass by. Most resin never becomes amber. In ordinary conditions, resin breaks down. It weathers, it oxidizes, it dissolves back into the environment, leaving no lasting trace. For resin to become amber, something different has to happen. It must be removed from the surface world. It must be buried. Over time, layers of sediment, sand, silt, mud cover the resin. Oxygen exposure decreases. Pressure slowly increases. Heat remains low but constant. Inside this quiet underground space, the chemistry of the resin begins to change. The small, volatile molecules that once gave fresh resin its scent gradually evaporate or rearrange. The remaining molecules begin to link together, forming long, stable chains. This process is called polymerization. It does not happen quickly. It unfolds over tens of thousands, hundreds of thousands, and sometimes millions of years. As polymerization continues, the resin becomes harder, more stable, less reactive. Eventually, it reaches a point where it no longer behaves like resin at all. At that stage, we call it amber. Amber is not frozen in time. It is transformed by time. What we hold today as amber is the result of slow chemical change, quiet burial, and deep patience. In the sections ahead, we'll follow this process more closely. We'll explore how amber forms, what it preserves, where it's found, and what it reveals about ancient forests and climates. But for now, it's enough to remember this simple beginning. Amber starts as a living response from a living tree, and everything that follows happens slowly beneath the surface, without urgency. Take a breath. Let your attention soften. We'll continue gently from here. To understand amber, it helps to stay for a moment with resin itself. Before resin becomes buried, before it changes chemically, before it becomes part of the geological record, it serves a role in the daily life of a tree. Resin is produced by certain types of trees, most commonly conifers such as pines, spruces, firs, and araucarias. Some flowering trees produce resins as well, though conifers are responsible for most of the amber deposits known today. Inside these trees, resin is stored in specialized channels. These resin ducts run through the wood and bark, forming a quiet internal network. Under ordinary conditions, resin remains contained, but when a tree is injured, that containment changes. Damage can come from many sources. Branches can snap under wind or snow. Animals can scrape bark while moving past. Insects can burrow into the wood to feed or lay eggs. Even temperature stress can cause small fractures to form. When this happens, resin begins to flow. The movement is slow and deliberate. Resin seeps outward rather than rushing. Its thickness allows it to cling to surfaces, spreading across exposed areas of the tree. This flow serves several functions at once. First, resin acts as a physical seal. It covers broken bark and exposed wood, reducing the entry of moisture. By limiting water access, the tree reduces the chance of rot. Second, resin contains chemical compounds that discourage microbial growth. Many resins are naturally antiseptic. Fungi and bacteria find it difficult to grow within it. Third, resin interferes with insects. Its stickiness can trap small arthropods that attempt to cross damaged areas. Its chemical makeup can also deter feeding or egg laying. In this way, resin functions as both barrier and signal. It isolates damaged tissue and stabilizes the tree's internal environment, while healing occurs beneath the surface. Resin is not produced continuously. It flows in response to need. Once a wound is sealed, resin production in that area slows or stops. The tree returns to a steady internal balance. In a forest setting, this process happens quietly and often invisibly. Trees experience minor injuries throughout their lives. Each response is localized, small in scale and temporary. Fresh resin reflects this purpose. When it first emerges, it is soft and pliable. It carries a strong aroma produced by volatile organic compounds that evaporate easily. These compounds play a role in defense as well. Some repel insects. Others inhibit microbial growth. Over time, they dissipate into the surrounding air. Resin's physical behavior also reflects its role. It can stretch into threads as gravity pulls it downward. It can pool in bark crevices. It can drip from branches and harden where it lands. In warm conditions, resin flows more readily. In cooler conditions, it thickens and slows. Seasonal temperature changes influence how far it spreads and how long it remains soft. In many forests, resin never leaves the tree. It hardens on the bark, forming glossy patches or small nodules. Rain, sunlight, and microorganisms gradually weather it away. Over months or years, it disappears. Sometimes, resin drips onto the forest floor. There, it may become coated with soil or leaf litter. It may break apart under pressure. It may be consumed or altered by microbes. This ordinary cycle represents the typical fate of resin. Amber forms only when this cycle is interrupted. But before that interruption occurs, resin's role remains biological rather than geological. It is part of a tree's immune response. It is temporary by design. It exists to solve a short-term problem. This perspective matters. Amber is often treated as a finished object, but its origin lies in a process meant to end. The resin that eventually becomes amber was never produced with permanence in mind. It was created to protect a living organism during a specific moment of stress. The forests that produced large amounts of resin were often dense, warm, and humid. In such environments, trees grew quickly and experienced frequent damage. Insects were abundant. Storms were common. High resin production was advantageous. These conditions help explain why certain geological periods produce more amber than others. They also help explain why amber preserves such intimate details of forest life. Resin flowed through ecosystems that were active, crowded, and biologically rich. Each drop interacted with its surroundings before it ever became buried. Understanding resin as a biological response grounds amber in the living world. Before amber becomes an archive of deep time, it begins as a brief, functional action taken by a tree in the present moment. In the next section, we'll follow what happens when that biological moment extends beyond its usual lifespan. We'll look at how resin leaves the surface world and enters a different environment entirely, where time begins to act in a new way. Resin becomes amber only when its usual story changes. Under ordinary conditions, resin remains part of the surface world. It hardens on bark. It drips onto soil. It weathers, cracks, and fades back into the environment. For amber to form, resin must leave this familiar setting. It must be buried. Burial does not happen all at once. It happens gradually, through the same quiet forces that shape landscapes everywhere. In forests near rivers, resin can be carried by flowing water. Floods move sediment, leaves, and organic debris together. Sticky droplets are lifted, rolled, and settled into low-lying areas. In coastal forests, resin may fall directly into shallow water. Because resin is less dense than water, it can float for a time, drifting along shorelines before becoming coated with fine sediment. In delta regions, mud and silt accumulate steadily. Each layer adds a little more weight. Each season contributes another thin covering. As resin becomes surrounded by sediment, its exposure to air decreases. Oxygen, which drives many chemical reactions at the surface, can no longer reach it easily. Microorganisms that normally break down organic material become less active. This change in environment marks an important transition. Resin is still organic material, but it is now isolated. Temperature beneath the surface remains relatively stable. It does not experience the daily swings of sunlight and cooling air. Heat is low but constant. Pressure increases very slowly. Not enough to crush or deform the material, but enough to influence its internal structure over long periods of time. Inside this buried setting, resin begins to change in a way that is almost imperceptible at first. Fresh resin contains many small molecules. Some are responsible for its scent. Others contribute to its softness and stickiness. These molecules are volatile, meaning they evaporate or react easily. Over long spans of time, these volatile components are gradually lost. They escape through microscopic spaces or rearrange internally. What remains is a more stable molecular framework. The molecules that stay behind begin to link together. Small chains connect to larger ones. Networks form. This slow linking process is known as polymerization. Polymerization does not involve melting. It does not involve crystallization. There is no dramatic transformation. Instead, the material becomes incrementally more rigid, incrementally more resistant to change. This process unfolds over tens of thousands of years, then hundreds of thousands, and sometimes millions. At first, the resin behaves much as it did before. It may still soften slightly with warmth. It may still respond to solvents. Over time, these responses diminish. The material grows harder. Its structure becomes more cross-linked. Its surface becomes smoother and more glass-like. Eventually, it reaches a point where it no longer behaves like fresh resin at all. At this stage, the material is recognized as amber. This transformation does not occur at a fixed moment. There is no single boundary where resin suddenly becomes amber. Instead, there is a gradual continuum. Younger material occupies an intermediate state. It retains some properties of resin, while beginning to take on the stability of amber. Older material shows the full expression of polymerization. It resists dissolution. It holds its shape. It persists. Throughout this entire process, the resin remains chemically organic. It does not convert into mineral matter. Its atoms are rearranged rather than replaced. This is why amber is classified as an organic fossil. Its preservation relies on isolation and chemical stability rather than mineral substitution. What makes this pathway remarkable is its reliance on patience rather than force. Amber does not require intense heat. It does not require extreme pressure. It does not require violent geological activity. It requires time, burial, and stillness. As sediment continues to accumulate above it, amber becomes part of the geological record. It may remain undisturbed for millions of years, protected within layers of rock. In some cases, later erosion exposes it again. Rivers cut into ancient deposits. Coastlines shift. Storms uncover buried layers. When amber returns to the surface, it carries with it a record of the environment in which it formed. But even then, its transformation remains ongoing. Amber continues to age slowly and subtly, long after it is removed from the ground. This quiet continuity is part of what makes amber unique. It is neither fully biological nor fully geological. It sits between these realms, formed by living processes, preserved by Earth systems. Understanding this pathway helps frame everything that follows. Amber is not simply resin that survived. It is resin that entered a different relationship with time. Next, we'll look more closely at what becomes enclosed within amber during this slow journey underground, and how that enclosure turns moments of forest life into lasting records. As resin continues along this slow path of burial and transformation, it does not change all at once. There is a long period during which the material exists in an in-between state. It is no longer fresh resin, but it has not yet reached the full stability of amber. This intermediate material is known as copal. Copal forms when resin has begun the process of polymerization, but has not experienced enough time, pressure, or chemical rearrangement to become fully fossilized. In practical terms, copal represents resin that is thousands to tens of thousands of years old, rather than millions. The distinction is based on behavior rather than appearance. At first glance, copal and amber can look remarkably similar. Both can be translucent. Both can display warm yellow, orange, or honey-colored tones. Both can contain trapped plant fragments or small organisms. The difference lies in how the material responds to its surroundings. Copal retains a closer chemical relationship to fresh resin. Its molecular chains are shorter. Its internal structure is less tightly linked. As a result, copal remains more reactive. When exposed to heat, copal softens more readily. When exposed to certain solvents, it can partially dissolve or become tacky. Its surface may scratch more easily under pressure. Amber, by contrast, reflects a more advanced stage of polymerization. Its molecular framework is more extensively cross-linked. This makes it harder, more stable, and less responsive to environmental changes. Heat affects amber more slowly. Solvents have limited effect. Its surface retains polish and structure over long periods. This difference explains why age alone is not always sufficient for classification. Two pieces of resin of similar appearance may behave very differently depending on how long and how deeply they were buried. In geological terms, time allows chemical rearrangement to continue. As volatile compounds are lost, the remaining molecular network becomes increasingly rigid. Each additional linkage reduces flexibility. Each rearrangement increases resistance. Over many thousands of years, copal gradually transitions toward amber. There is no sharp boundary where this transformation completes. Instead, the material moves along a spectrum. Some samples display characteristics of both stages. They may resist solvents but soften under sustained warmth. They may appear solid but retain faint resinous scent when heated. These transitional pieces offer insight into the process itself. They show that amber formation is not an event, but a progression. Environmental conditions play a role in how quickly this progression unfolds. Resin buried beneath thick, stable sediment tends to mature more consistently. Limited oxygen exposure slows degradation. Stable temperatures support gradual polymerization. Resin that remains closer to the surface or experiences repeated exposure to air and moisture may never advance beyond the copal stage. This is one reason amber deposits are relatively rare. They require not only resin production, but also the right conditions for preservation over extended time. The distinction between copal and amber also matters for scientific study. Amber offers long-term chemical stability. It can preserve delicate internal structures for millions of years. Copal, while still informative, is more vulnerable to alteration. Microstructures may shift. Chemical signals may degrade. Long-term preservation is less certain. Understanding this difference helps researchers interpret what they observe. It also helps explain why amber has played such a central role in reconstructing ancient forest environments. Amber represents resin that completed the full journey. Copal represents resin still in transit. Both are part of the same continuum. In everyday handling, this continuum becomes tangible. Copal often feels slightly warmer to the touch. It may appear softer under pressure. Its surface may show subtle changes over time. Amber, by contrast, maintains its form. It holds polish. It persists through centuries of handling and exposure. This durability is what allowed amber to survive long enough to be incorporated into sedimentary rock and later rediscovered. It is also what allows amber to function as a long-term archive. The difference between copal and amber is not a matter of value or quality. It is a reflection of time. Each represents a moment along the same pathway, shaped by burial depth, chemistry, and patience. By recognizing this gradual transition, the story of amber becomes clearer. Resin does not leap from tree to fossil. It moves forward slowly, changing incrementally as its relationship with the environment shifts. As this transformation continues, anything enclosed within the resin becomes part of that journey as well. Resin continues to change beneath layers of sediment. Time becomes the primary influence acting upon it. Amber can be very old. In some rare cases, amber dates back more than 300 million years to forests that existed long before flowering plants appeared. These early ambers formed during the Carboniferous period, when Earth's landscapes were dominated by vast, swampy forests and atmospheric conditions differed greatly from today. Such ancient amber is uncommon. It survives only where burial conditions remain stable for immense spans of time. Even slight disruption over hundreds of millions of years can prevent resin from completing its transformation. More commonly, amber deposits are younger, formed during the age of dinosaurs and afterward. Many of the most scientifically rich amber deposits date to the Cretaceous period, roughly 100 million years ago. During this time, forests were widespread, warm, and biologically active. Resin production was frequent, and sedimentary environments allowed for consistent burial. Other major deposits formed later, during the Paleogene and Neogene periods. These include ambers that are tens of millions of years old, rather than hundreds. Age matters, but it does not act alone. The age of amber reflects a balance between time and preservation. Resin may be produced continuously in forests, but only a small fraction ever becomes buried deeply enough to escape weathering and decay. Once buried, it must remain undisturbed. Erosion, tectonic movement, heat, or prolonged exposure to oxygen can interrupt the slow chemical changes required for amber to stabilize. Because of this, amber does not appear evenly across the geological record. It clusters in specific intervals, marking times when forests, climate, and sedimentary conditions aligned. Dating amber relies on the rock layers surrounding it. Amber itself does not contain minerals suitable for radiometric dating. Instead, scientists determine its age by examining the sediments in which it is found. Fossils of plants and animals within those layers provide additional context. Volcanic ash layers, when present nearby, offer precise time markers. Through this approach, amber becomes part of a broader geological timeline. Some amber deposits formed in ancient coastal environments. Resin fell into shallow water, floated briefly, and then settled as fine sediments accumulated around it. Other deposits formed inland, within river floodplains or forest soils. Seasonal flooding buried resin repeatedly, layer upon layer. Each environment influenced the final character of the amber. Amber formed in marine settings often shows signs of transport. Its surface may be smoothed by gentle movement. Inland amber may retain sharper contours, reflecting shorter travel before burial. Age also affects the internal chemistry of amber. Over millions of years, polymerization continues at a slow pace. Molecular networks become more tightly linked. Chemical reactions that proceed quickly at the surface stretch out across geological time. This ongoing stabilization contributes to amber's durability. Older amber tends to resist chemical change more effectively. Its structure becomes increasingly inert. This does not mean younger amber is incomplete. It reflects a different stage along the same pathway. Amber from different ages can preserve biological material with remarkable clarity. The key factor is isolation rather than age alone. Once resin is sealed away from oxygen and microbes, even relatively young amber can preserve fine details. Age becomes especially meaningful when amber is viewed as a record. Each deposit captures a specific moment in Earth's history. Not a single day or season, but a span of time during which resin production and burial were active. Amber from one region may record a humid tropical forest. Amber from another may reflect a coastal woodland. Differences in age allow scientists to compare ecosystems across eras. This comparison reveals how forests changed through time, which plants dominated, which insects thrived, how climates shifted. Amber does not provide a continuous record. It offers windows. Each window opens onto a different world, separated from the present by millions of years. Holding a piece of amber is holding that separation. It is a material that has persisted through continental movement, through climate shifts, through extinction and renewal. Its age is not marked on its surface, but embedded within its structure and the rocks that cradle it. Time acts quietly on amber, leaving no obvious signs of struggle or force. What remains is a stable form, carrying the imprint of the conditions that allowed it to endure. As amber continues to be uncovered and studied, its age helps place each piece within the larger story of Earth, not as an isolated object, but as part of a long, uneven timeline shaped by forests, sediments, and patience. As resin continues to mature beneath layers of sediment, its transformation is guided by chemistry that unfolds slowly and quietly. The substances that make up fresh resin are complex, but their behavior can be understood without detail or formula. At its origin, resin is composed largely of organic molecules known as terpenoids. These molecules are produced by plants as part of their normal biological processes. They are lightweight, reactive, and well suited to movement and change. Terpenoids give fresh resin its familiar qualities. They contribute to its aroma. They allow it to remain fluid or sticky. They enable it to respond quickly to temperature and air. When resin is newly formed, these molecules exist largely as small, separate units. They move freely within the resin, interacting easily with their surroundings. Once buried, that freedom begins to diminish. Over long periods of time, the smallest and most volatile components gradually disappear. Some evaporate slowly through microscopic spaces. Others rearrange internally as chemical bonds shift. As these lighter components are lost, the remaining molecules begin to interact more closely. Individual terpenoid units link together. Short chains form, then longer ones. These chains do not align in neat rows or repeating patterns. Instead, they connect irregularly, branching and crossing in multiple directions. Over time, this growing network becomes more extensive. What begins as a collection of small molecules becomes a complex, interlinked structure. This cross-linked network is what gives amber its stability. Rather than behaving like a soft, flowing substance, amber behaves like a solid framework. It does not melt under modest heat. It does not dissolve easily. Its shape remains consistent over long periods. This structure is sometimes described as a natural plastic, not in the modern, manufactured sense, but in the way it combines flexibility at the molecular level with durability at the material level. The comparison is useful because it emphasizes structure over origin. Amber's strength comes from how its molecules are connected, not from mineral hardness or crystal arrangement. As polymerization continues, the network becomes increasingly resistant to change. Chemical reactions that once occurred readily slow to a near standstill. The material becomes chemically inert. This inertness is what allows amber to persist. It limits interaction with water. It resists microbial activity. It maintains its internal composition. One way to imagine this change is to picture individual strands slowly weaving into a net. At first, the strands are loose. They shift and move. Openings remain wide. With time, more connections form. The net tightens. Movement becomes restricted. The structure holds. Another subtle change occurs during this process. Fresh resin carries a strong scent because it contains volatile aromatic compounds. These compounds are among the first to be lost during burial. As they fade, the resin's interaction with air diminishes. Older amber retains only a faint scent when warmed. This lingering aroma reflects the last traces of those original compounds trapped within the hardened network. In certain amber deposits, the chemical signature includes compounds that attract particular attention. Baltic amber, for example, often contains measurable amounts of succinic acid. This has led to the historical term succinite being applied to amber from this region. Succinic acid does not define amber as a whole. Its presence reflects the chemistry of the trees that produce the resin and the conditions under which it matured. Different forests produced resins with different chemical balances. Different burial environments influenced how those chemicals evolved. As a result, amber from different regions can vary slightly in composition while sharing the same fundamental structure. These variations help scientists trace amber back to its source. Chemical profiles act as subtle fingerprints, linking amber to specific plant groups or environments. Despite these differences, the overall behavior of amber remains consistent. Its cross-linked polymer network resists decay. Its chemical inertness protects what it encloses. Its structure remains stable across immense spans of time. This stability explains why amber responds differently to heat and solvents than younger resins. Where copal may soften or dissolve, amber maintains its form. The chemistry that once allowed resin to flow and heal wounds has reorganized into something far more enduring. Yet even in its hardened state, amber carries traces of its origin. Its warmth to the touch, its subtle glow, and its faint scent when heated all reflect its biological beginnings. Amber is neither fully biological nor fully geological. Its chemistry occupies a middle ground. It is shaped by living processes, then preserved by Earth systems through molecular rearrangement rather than replacement. Understanding this chemistry provides a foundation for what amber can hold. Its stability is not incidental. It is the reason amber can preserve delicate forms that would otherwise disappear. Within this hardened network, details remain suspended, protected from time by the quiet order of linked molecules. The chemistry of amber creates the conditions for something remarkable to persist. One of the most distinctive qualities of amber is what it carries within it. As resin flows through a forest environment, it moves through air filled with particles. Pollen drifts invisibly between trees. Fungal spores circulate close to the ground. Fragments of leaves and bark fall continuously. When resin encounters these elements, it does not filter or select. It encloses whatever it touches. This makes amber a record of everyday forest life, captured at a very small scale. Among the most common inclusions in amber are pollen grains. These microscopic structures are released by plants in enormous numbers. Their shapes vary by species, and their outer walls are remarkably resilient. When pollen becomes enclosed in resin, it retains its form with extraordinary clarity. Fine surface textures remain visible. Spines, pores, and ridges persist. Because pollen is produced seasonally, its presence in amber provides clues about flowering cycles and forest composition. Alongside pollen, amber often preserves fungal material. Delicate filaments, known as hyphae, can be found suspended within the hardened resin. Fungi play a central role in forest ecosystems. They decompose organic matter, form relationships with plant roots, and circulate nutrients through soil. Amber captures fragments of this hidden network, preserving evidence of interactions that rarely fossilize in other ways. Plant material is also common. Tiny leaves, needles, seeds, and fragments of bark appear frequently in amber deposits. Some are caught mid-fall. Others rest against the resin's surface, suggesting brief contact before enclosure. Even the cellular structure of these plant fragments can remain intact. Cell walls, veins, and growth patterns are sometimes visible under magnification. Insects are among the most recognized inclusions in amber. Small flies, ants, beetles, and wasps are frequently found suspended within it. Their exoskeletons, wings, antennae, and eyes are preserved in 3 dimensions. Unlike compression fossils, which flatten organisms into thin outlines, amber maintains spatial form. This allows fine anatomical details to persist. Wing veins remain layered. Compound eyes retain individual facets. Mouthparts and joints remain articulated. Beyond insects, amber also preserves other small arthropods. Mites, spiders, and ticks appear regularly. Some are found attached to plant material. Others appear alone, suggesting movement through the forest air. In rarer cases, amber preserves fragments of vertebrates, small feathers, hair, skin fragments, and occasionally portions of tails or limbs have been identified. These finds are uncommon, but they extend amber's record beyond invertebrate life. What unites all of these inclusions is scale. Amber does not record entire landscapes. It records moments. It captures what happened to be present within a few centimeters of flowing resin. This limitation is also its strength. Amber preserves fine detail that larger-scale fossils cannot. The structures it encloses are often too small, too delicate, or too transient to survive burial in sediment. Because amber seals its contents rapidly, it limits exposure to oxygen and microbes. Decomposition slows dramatically. Tissues do not collapse. Surfaces remain intact. In some cases, even internal structures persist. Air bubbles trapped within resin can outline soft tissues. Surface tension preserves posture. The orientation of limbs or wings reflects the moment of contact. This level of preservation is sometimes described as microscopic fidelity. It does not imply perfection. It reflects resolution. Amber preserves detail at the scale of millimeters and micrometers, offering a view into aspects of ecosystems that are otherwise invisible in the fossil record. These details allow scientists to examine relationships as well as individuals. An insect covered in pollen suggests plant interaction. A parasite attached to a host suggests behavioral patterns. Fungal spores on plant fragments suggest decomposition already underway. Each inclusion provides context. Rather than isolated specimens, amber often preserves small ecological scenes. These scenes are incomplete, but they are informative. They reveal how organisms coexisted, how they interacted, and how they occupied space within the forest. Because amber forms from resin that flowed in specific locations, its record reflects the structure of forests themselves. Canopy insects, ground-dwelling arthropods, and organisms living on bark are more likely to appear than species inhabiting open water or deep soil. This bias is important to recognize. Amber does not represent everything. It represents what resin encountered. Even so, the range of life preserved within amber is broad. It includes producers, consumers, decomposers, and parasites. It spans plants, fungi, invertebrates, and fragments of vertebrate life. Together, these inclusions create a layered picture. Not a snapshot of a single organism, but a textured glimpse of a living system, held within a stable, transparent material. Amber does not isolate life from its environment. It carries environment with it. Within its hardened structure, the small details of ancient forests remain present, quietly preserved by chemistry and time. When resin flows through a forest, it does not act with intention. It responds to gravity, temperature, and surface texture. Its movement is slow but persistent. The process by which organisms become enclosed in resin unfolds through a series of simple physical steps. It begins with contact. Fresh resin is viscous and adhesive. When a small organism touches it, movement becomes difficult immediately. Insects that land on resin-coated bark often struggle to free themselves. Legs adhere first. Wings follow. For organisms moving along branches or trunks, resin behaves like a sudden change in terrain. What was once a solid surface becomes soft and resistant. As the organism attempts to move, it presses deeper into the resin. This contact increases surface area. More of the body becomes coated. Escape becomes less likely. The next step involves continued resin flow. Resin rarely emerges as a single release. It often flows intermittently, especially if damage to the tree remains open. Additional resin may ooze over the original flow, covering what is already present. This secondary coating is important. It seals the organism more completely, reducing exposure to air. As resin layers accumulate, they isolate the enclosed material from the surrounding environment. Air pockets may remain at first. Small bubbles can form around body surfaces. These bubbles outline shape and preserve spacing between structures. Over time, resin begins to firm at the surface. The outer layer hardens first, forming a skin. Beneath this skin, the interior remains softer for longer. This gradual hardening limits movement. The organism becomes fixed in position. Posture at the moment of enclosure is often preserved. Wings may be extended, legs may be bent or stretched, antennae may curve gently. Once the resin fully hardens, the enclosed material is sealed. From this point onward, chemical isolation begins. Oxygen availability drops sharply, microbial activity slows. Decomposition, which relies on oxygen and moisture, becomes limited. Soft tissues may collapse partially, but surface features remain. Exoskeletons retain form. Plant structures remain layered. If burial follows, this sealed resin enters a new phase. Sediment accumulates around it. Pressure increases slowly. Temperature stabilizes. The resin no longer interacts with the surface environment. This isolation protects what lies within. Over time, polymerization strengthens the surrounding material. The enclosure becomes more rigid and stable. The enclosed organism does not fossilize in the traditional sense. Its tissues are not replaced by minerals. Instead, they remain suspended within a hardened organic matrix. This difference explains why amber inclusions often appear lifelike. They retain volume. They preserve spatial relationships. Details such as eye facets, wing veins, and surface textures remain visible. The step-by-step nature of this process means that enclosure does not require sudden events. A forest does not need catastrophe to produce amber inclusions. Routine injury to trees, combined with ordinary movement of organisms, is enough. Small interactions, repeated many times, produce the record. Not every contact results in preservation. Some organisms escape before sealing occurs. Some resin flows dry too quickly. Some are disrupted before burial. The inclusions that remain represent the subset of encounters that aligned with timing and conditions. This selective process shapes what amber contains. Organisms active on bark, leaves, and branches are more likely to be preserved than those living underground or in open water. Lightweight organisms encounter resin more often than heavier ones. Even within these limits, the range of preserved life is wide. The process remains consistent across forests and eras. Contact, adhesion, additional flow, sealing, burial. Each step builds upon the last, without intention or urgency. Amber forms through accumulation, not intervention. What remains is a quiet record of interactions that once lasted only moments, now extended across deep time by chemistry and patience. Amber holds moments. Not moments in the human sense of memory, but moments of physical arrangement. A wing mid-beat. A grain of pollen suspended in air. A fragment of bark drifting downward. When resin encloses these details, it fixes a specific configuration of space and matter. Nothing inside amber is staged. Nothing is prepared. The material records whatever happened to be present at the instant contact occurred. This quality gives amber a unique role in the geological record. Most fossils represent extended processes. Bones settle, compress, mineralize. Shells accumulate layer by layer. Impressions form through repeated pressure. Amber works differently. It preserves brief encounters, an insect landing, a spore drifting, a feather brushing against flowing resin. These events may have lasted only seconds, yet their physical arrangement becomes stable for millions of years. This contrast between duration and persistence is what allows amber to function as a time capsule. One of the most striking examples of this preservation comes from amber dating to approximately 99 million years ago. Within this amber, a portion of a small dinosaur's tail was discovered, complete with feathers. The specimen does not represent a skeleton. It does not show the full animal. It preserves a fragment, caught during ordinary contact with resin. The feathers remain layered. Barbs and filaments are visible. Soft tissue outlines can be traced. This find demonstrated something important. Amber is capable of preserving details that rarely survive in other fossil contexts. Feathers, skin, hair, and other delicate structures are typically lost early in fossilization. In amber, these structures can persist. The value of this preservation lies not in rarity alone, but in resolution. Amber captures fine-scale anatomy. It preserves surface relationships. It maintains three-dimensional form. These qualities allow scientists to examine structures as they existed in life, rather than as flattened or mineral-replaced remnants. Even when the organism itself is incomplete, the preserved portion retains context. Feather arrangement reveals insulation or display patterns. Hair density suggests thermoregulation. Skin texture reflects surface adaptation. Beyond dramatic examples, most amber inclusions are far smaller. A fly caught mid-step, a spider suspended between movements, a mite clinging to a plant hair. These modest scenes are equally informative. They reveal posture, orientation, and interaction. They show how bodies occupied space. They capture behavior without narration. A parasite attached to its host records a relationship. An insect coated in pollen records contact with flowering plants. Each inclusion is static, but the implications are dynamic. Amber does not freeze motion in a cinematic sense. It stabilizes a physical arrangement. What remains is a configuration that can be examined long after movement has ceased. This stabilization allows amber to bridge biology and geology. It preserves biological detail within a geological material. The enclosing resin transforms into amber, but the internal scene remains insulated from that transformation. This separation between container and contents is essential. Amber hardens and stabilizes while maintaining a protective boundary. Within that boundary, fine structures persist. The moment does not decay further. Amber does not preserve sound or light or intention. It preserves form. That form is enough. By studying these preserved forms, scientists reconstruct aspects of ancient ecosystems that would otherwise be inaccessible. They infer behavior, movement, and interaction. They see not just what existed, but how it was arranged. Each piece of amber holds a limited view, but across many specimens, patterns emerge. Forests reveal themselves through repeated small moments. Amber's role as a time capsule rests on this accumulation. Not a single dramatic event, but thousands of ordinary interactions held long enough to be studied. What amber offers is not a story told after the fact, but a moment carried forward, unaltered in its physical arrangement, through deep time. Amber forms under particular environmental conditions, and those conditions leave traces within it. Resin production is not evenly distributed across climates. Trees produce more resin when certain factors align. Warm temperatures increase resin flow. Moist environments support dense vegetation. Insect activity rises in such settings, leading to more frequent damage to bark and branches. Together, these factors create forests where resin production is sustained. When amber deposits appear in the geological record, they often coincide with periods characterized by warmth and humidity. These climates support rapid plant growth. Trees experience repeated stress from storms, heavy rainfall, and biological activity. Resin becomes a frequent response. As a result, large amounts of resin enter surrounding environments. Amber therefore serves as an indirect indicator of ancient climate. It does not measure temperature directly. It reflects the conditions that favored resin production and preservation. Many amber deposits are associated with coastal forests, river deltas, and low-lying floodplains. These environments combine high biological productivity with steady sediment accumulation. In coastal regions, trees grow close to shorelines and estuaries. Resin that drips from branches may fall directly into shallow water. Because resin is less dense than water, it can float. Floating resin may drift along calm shorelines before becoming coated with fine particles. As sediment settles, resin becomes heavier. It sinks and is incorporated into the seabed. In river systems, seasonal flooding plays a similar role. Floodwaters transport sediment across forest floors. Resin droplets and nodules are lifted and carried short distances. When waters recede, sediment settles around them. Repeated flooding buries resin incrementally, creating layered deposits. Lagoonal environments provide another pathway. Sheltered bodies of water accumulate fine sediments at a steady rate. Resin introduced into these settings is quickly surrounded and isolated. These depositional settings share important characteristics. They limit exposure to oxygen. They reduce mechanical disturbance. They promote gradual burial. As amber forms within these environments, it carries with it additional environmental information. Pollen trapped in amber reflects surrounding plant communities. The types of insects preserved suggest temperature and humidity ranges. Certain insect groups thrive in warm, moist conditions. Their presence within amber supports climatic interpretation. Chemical signatures within amber also offer clues. The composition of resin reflects the species of trees present. Different trees dominate under different climatic regimes. By identifying resin chemistry and associated inclusions, scientists infer forest structure and climate. Amber does not record climate in isolation. It records climate through ecosystem response. Dense forests, high insect diversity, and frequent resin flow are expressions of environmental conditions. Changes in amber abundance over time reflect shifts in these conditions. Periods with abundant amber deposits often correspond to intervals of global warmth. Periods with fewer deposits suggest cooler or drier climates, or environments less favorable to resin preservation. These patterns are broad rather than precise. Amber does not resolve short-term fluctuations. It integrates conditions across long spans of time. This integration smooths variability, highlighting persistent trends. Amber also captures evidence of disturbance. Storms, floods, and shifting shorelines leave their mark through transport and burial patterns. Resin pieces that show rounding or abrasion suggest movement through water. More angular pieces suggest limited transport and rapid burial. By studying these features, scientists reconstruct how resin moved through landscapes before becoming amber. Climate, hydrology, and sedimentation work together in this process. None act alone. Amber emerges where biological productivity meets geological opportunity. The forests that produced amber were not static. They responded to changing rainfall, sea levels, and temperature. Amber preserves evidence of these responses in subtle ways. The material itself records chemistry. The inclusions record life. The surrounding sediments record environment. Together, they form a layered archive. Through amber, ancient climates are not described directly, but inferred through the quiet accumulation of biological and geological signals. These signals persist long after the forests that produced them have disappeared. Amber remains as their trace, carrying the imprint of warmth, moisture, and the ecosystems shaped by them. Once resin enters the environment beyond the tree, its future depends largely on where it comes to rest. Amber does not form everywhere resin falls. It accumulates in particular places, shaped by gravity, water movement, and sediment. These locations share one essential quality. They allow resin to settle and remain undisturbed long enough for burial to begin. Shorelines are one such place. In coastal forests, trees often grow close to water. Branches extend over tidal flats, estuaries, and shallow seas. When resin drips from bark or breaks free from branches, it may fall directly into water. Because resin is buoyant, it can float for a time at the surface. While floating, it drifts slowly, guided by gentle currents and tides. It may travel only short distances before encountering suspended sediment. Fine particles in the water adhere to the sticky surface of the resin. As these particles accumulate, the resin becomes heavier. Eventually, it settles. Once on the seabed, the resin rests among sand, silt, and organic debris. Additional sediment continues to fall, covering it layer by layer. This gradual covering shields the resin from disturbance. River floodplains provide another pathway. Forests near rivers experience regular flooding. During high water, sediment spreads across low-lying ground. Resin droplets and fragments lying on the forest floor are lifted briefly by moving water. They shift position, often only a short distance. When floodwaters recede, sediment settles. Resin becomes embedded within layers of mud and silt, sometimes buried in a single event, sometimes over many seasons. Floodplains build upward slowly. Each flood adds a thin layer. Each layer increases isolation. Over time, resin trapped in these settings moves deeper below the surface. Lagoonal environments are especially effective at preserving resin. Lagoons are shallow bodies of water, often separated from open seas by barriers or reefs. Water movement is limited. Sediment accumulates steadily. Resin introduced into lagoons is quickly surrounded by fine-grained material. This rapid burial limits exposure to oxygen and reduces mechanical stress. Because lagoons are often rich in organic matter, they also support diverse plant and insect life, increasing the likelihood that resin carries inclusions. Inland forest soils can also contribute to amber formation, though less consistently. Resin that hardens on the forest floor may be covered by leaf litter and soil. Burial in these settings depends on stability. If erosion is minimal and sediment continues to accumulate, resin may remain protected. More often, forest floor resin is disturbed or degraded before long-term burial can occur. This variability helps explain why amber deposits are unevenly distributed. Some regions contain abundant amber. Others, despite having resin-producing trees, contain very little. The difference lies not in resin production alone, but in the presence of environments that collect and preserve it. Once resin becomes part of a sedimentary deposit, its position relative to surrounding materials matters. Resin tends to concentrate in specific layers, often associated with organic-rich sediments. These layers may be thin, but they can extend across wide areas. Over time, tectonic movement, erosion, and sea level change can expose these layers again. Rivers cut into ancient deposits. Coastlines retreat or advance. Sedimentary layers tilt or fracture. Amber reappears at the surface, sometimes far from where it originally formed. Beach deposits of amber often originate this way. Eroded material is carried by water, sorted by size and density. Amber, being light, can accumulate along shorelines where waves deposit organic debris. These secondary deposits represent amber that has traveled, sometimes far, from its original resting place. By studying where amber collects, scientists reconstruct the pathways resin followed from forest to sediment. They examine grain size, associated fossils, and layering patterns. These physical clues reveal how landscapes shaped preservation. Amber formation depends on quiet accumulation rather than force. It favors low-energy environments where sediment settles gently. It reflects places where time was allowed to work without interruption. Through shorelines, floodplains, and lagoons, resin finds stillness. That stillness allows transformation. Amber collects where movement slows, where particles gather, and where burial becomes possible. The places amber rests become part of its story, holding it long enough for time to take over. Amber does not appear evenly across the earth. It gathers in particular regions where forests, climate, and sediment aligned long enough for resin to complete its slow transformation. Some of these regions have produced far more amber than others, not because trees were unique, but because preservation conditions remain stable over long spans of time. The largest and most widely known amber deposit comes from the regions surrounding the Baltic Sea. Baltic amber formed during the late Eocene period, roughly 34 to 38 million years ago. At that time, Northern Europe supported extensive forests dominated by resin-producing trees. The climate was warmer and more humid than it is today. Resin flowed frequently, and rivers carried it toward shallow marine environments. There, fine sediments accumulated steadily. This combination allowed large volumes of resin to be buried, preserved, and transformed. Today, Baltic amber is found along coastlines and within sedimentary layers across countries bordering the Baltic Sea. Its abundance has made it a central reference point for understanding amber as a material and as a record of ancient forest ecosystems. Another important deposit lies in what is now northern Myanmar. This amber formed much earlier, during the mid-Cretaceous period, approximately 99 million years ago. At that time, the region supported tropical forests rich in biological diversity. Myanmar amber is notable for its age and for the range of organisms preserved within it. Because it formed during the age of dinosaurs, it captures forest life from a very different world. The resin-producing trees were part of ecosystems that no longer exist in the same form. As a result, the inclusions found in this amber often represent extinct lineages, providing insight into early evolutionary relationships. In the Caribbean, amber deposits formed much more recently. Dominican amber dates to roughly 15 to 20 million years ago, during the Miocene epoch. These deposits originated in tropical forests on islands influenced by volcanic activity and shifting coastlines. Resin flowed from trees growing near rivers and coastal plains, and burial occurred within sediment-rich environments. Dominican amber is especially clear, allowing fine internal details to be seen easily. Its clarity has made it valuable for studying insects, plants, and ecological interactions in ancient tropical forests. Nearby regions in Mexico contain amber of similar age and origin. These deposits reflect related forest systems, shaped by similar climatic and geological conditions. Beyond these well-known regions, amber appears in many other parts of the world. Deposits have been identified in parts of Spain and France, where ancient coastal forests once stood. Amber is found in Lebanon, representing some of the oldest known fossil resins, formed during the early Cretaceous period. In North America, amber occurs in regions such as New Jersey and Canada, often associated with ancient coastal plains. Smaller deposits appear in Russia, China, and parts of Southeast Asia. Each deposit reflects a local story, the type of trees present, the nature of surrounding sediments, and the pace of burial all influence the character of the amber that forms. Some ambers are opaque. Others are remarkably clear. Some contain abundant inclusions. Others preserve only plant fragments or pollen. Together, these deposits create a global pattern. Amber appears where forests once thrived, where water carried sediment gently, and where landscapes allowed stillness. By mapping these deposits, scientists trace the distribution of ancient forests across continents and eras. Amber becomes more than a material. It becomes a geographic record, marking where life and land intersected in ways that allowed time to work slowly and completely. Each region holds a different chapter, preserved not in stone, but in hardened resin that traveled quietly through time. The places where amber is found today do not always match where it first formed. Forests shift, land moves, coastlines change. Over deep time, the surface of the earth is not fixed. Amber deposits are shaped not only by local environments, but by the slow movement of continents. This movement is driven by plate tectonics. The Earth's outer shell is divided into large plates that drift gradually across the planet's surface. Their motion is measured in centimeters per year, but over millions of years, those small movements add up. Forests that once grew near the equator may now sit at higher latitudes. Coastlines that once bordered shallow seas may now lie far inland. Amber records these journeys. Many amber deposits formed in regions that occupied very different positions on the globe at the time of resin production. Baltic amber provides a clear example. When the resin that became Baltic amber was flowing, the land that now surrounds the Baltic Sea was positioned farther south than it is today. The climate was warmer, supporting dense resin-producing forests. As tectonic plates shifted northward, those forests disappeared, but the amber remained buried within sediment. Over time, erosion and geological uplift exposed those layers again, revealing amber far from the environment in which it formed. Myanmar amber reflects an even longer journey. During the mid-Cretaceous period, the land that now forms northern Myanmar was part of a complex tectonic setting. Fragments of continental crust moved independently, colliding and merging over millions of years. Resin formed in tropical forests on land masses that were still in motion. As plates converged, sediments were compressed and altered. Amber remained protected within them, carried along as continents assembled. By the time the amber was exposed, its original forest had long vanished, and its position on the globe had changed dramatically. Amber deposits in the Caribbean also reflect tectonic movement. Island arcs formed through volcanic activity as plates interacted beneath the ocean. Forests grew on newly emerged land, producing resin that was buried in nearby sediments. As plates continued to shift, these islands moved relative to one another, reshaping coastlines and sedimentary basins. Amber formed in one setting and later emerged in another. This pattern appears repeatedly around the world. Amber found in Spain and France formed when those regions lay closer to ancient coastlines. Deposits in North America record forests that existed along shifting continental margins. In each case, the amber did not move on its own. The land beneath it moved. Plate tectonics also influences where amber survives. Mountain building can lift sedimentary layers, bringing amber closer to the surface. Erosion can remove overlying rock, freeing amber from long burial. In other cases, tectonic activity buries amber too deeply, subjecting it to heat and pressure that alter or destroy it. The presence of amber today reflects a balance between movement and protection. Too much disturbance prevents preservation. Too little prevents exposure. The amber that remains marks places where geological forces acted gently enough to allow resin to endure. This connection between amber and plate tectonics adds depth to its story. Amber is not only a product of forests and chemistry, it is also a traveler, carried across the planet by the slow rearrangement of land. When amber is studied in context, it reveals both biological history and geological motion. Its chemistry speaks of trees. Its inclusions speak of ecosystems. Its location speaks of moving continents. Together, these elements show how life and Earth shape one another over deep time. Amber does not belong to a single place. It belongs to the long movement of the planet itself, preserved quietly as the ground beneath it drifted, shifted, and changed. Long before amber was studied scientifically, it was noticed, handled, and carried. People encountered amber where it emerged naturally, along shorelines, within eroded sediments, among organic debris washed up by water. Its appearance set it apart. Amber is light. It warms quickly in the hand. It glows softly when held to the light. These qualities invited attention. Some of the earliest known uses of amber date back more than 11,000 years. Small amber beads and pendants have been found in prehistoric sites across Europe. They were shaped with simple tools, polished through handling, and worn close to the body. These early uses suggest familiarity rather than rarity. Amber was gathered where it appeared, worked where it was found, and incorporated into daily life. As human societies became more complex, amber continued to circulate. In ancient Egypt, amber was valued for its appearance and symbolism. It was used in jewelry and placed in tombs, often alongside other organic materials. Egyptian texts and artifacts reflect an association between amber and the sun, likely inspired by its color and glow. In Northern Europe, amber became part of long-distance exchange networks. Material from the Baltic region traveled far beyond its place of origin. It moved southward along river systems and across land routes. This movement occurred gradually, over generations, rather than through sudden expansion. Amber did not function solely as ornament. In many cultures, it was attributed protective or restorative properties. When rubbed, amber develops a static charge. This subtle effect was noticed early and interpreted in various ways. The ancient Greek word electron was used to describe amber, later giving rise to the term electricity. This physical response contributed to amber's symbolic meaning, linking it to unseen forces and energy. Amber was also burned as incense in some traditions. When heated, it releases a faint resinous scent, a remnant of its botanical origin. This scent reinforced its association with trees, forests, and living landscapes. In medieval Europe, amber continued to appear in religious and decorative contexts. It was carved into beads, rosary components, and small objects. Trade routes shifted, but amber remained present. In some regions, its use became regulated. Control over amber collection reflected its cultural importance and its association with coastal territories. Alongside symbolic meanings, amber was sometimes used for practical purposes. Powdered amber appeared in historical remedies, often connected to the presence of succinic acid, particularly in Baltic amber. These practices reflected the medical understanding of the time rather than measured effectiveness. The belief that amber contained beneficial properties persisted across cultures. What is consistent across these uses is continuity. Amber was never confined to a single period or a single interpretation. Its meaning adapted as societies changed. Unlike metals or stone, amber retained its organic identity. It remained recognizable across centuries, even as techniques and beliefs evolved. This continuity mirrors amber's origin. It began as a biological response, entered the geological record, and later re-entered human experience without losing its material character. Amber's role in human history is not defined by sudden discovery or transformation. It reflects long familiarity. People lived alongside amber wherever it emerged from the ground. They handled it, wore it, and passed it along. In this way, amber connects deep time to lived time. It links ancient forests to human hands. The same material shaped by chemistry and burial became part of ritual, adornment, and trade. Amber did not need explanation to be valued. Its presence was enough. Across cultures and eras, it remained a quiet companion, carried forward much like the moments it preserves within itself. Amber has qualities that are immediately noticeable the moment it is picked up. It feels different from stone. When held in the hand, amber warms quickly. It does not retain the cool temperature of minerals or metal. Its warmth reflects its organic origin, the way it interacts with heat rather than resisting it. This tactile response has shaped how people perceive amber across cultures and time. Amber is relatively light. Compared to stone of similar size, it weighs noticeably less. This low density comes from its molecular structure. The cross-linked polymer network that gives amber stability also leaves small spaces within the material. These spaces reduce overall mass without compromising form. One way this lightness becomes apparent is when amber is placed in salt water. In a concentrated salt solution, amber floats. This behavior distinguishes it from many minerals and reflects its resinous beginnings. The ability to float also influenced how amber moved through ancient environments, allowing resin fragments to travel across shallow waters before burial. Amber's hardness is modest. On the Mohs scale of mineral hardness, amber ranks around 2. This means it can be scratched by harder materials, but it also means it can be shaped with relative ease. Early craftspeople worked amber using simple abrasion and polishing techniques. The material responds smoothly to shaping. Edges soften rather than fracture. Surfaces become glossy through repeated contact. This workability contributed to amber's long history of use in beads, pendants, and small objects. Color is one of amber's most striking features. Although often associated with golden or honey tones, amber appears in a wide range of colors. Yellows, oranges, and browns are common. Reds and deep cognac shades occur as well. Some amber appears pale or milky. Others range toward green or nearly black. These variations reflect differences in resin composition, burial conditions, and the presence of microscopic inclusions or bubbles. Clear amber allows light to pass through easily, revealing internal structures. Opaque amber scatters light, creating a soft glow rather than transparency. Both forms reflect the same material, expressed differently through chemistry and history. Amber interacts with light in a gentle way. Its refractive qualities create depth, giving the impression that objects within it are suspended rather than trapped. This visual softness contributes to its appeal and distinguishes it from glass or crystal. Another subtle property of amber emerges when it is rubbed. Friction causes amber to develop a static electric charge. This effect is mild but noticeable. Small particles such as dust or hair may be attracted briefly to its surface. This phenomenon was observed in antiquity and recorded long before electricity was understood. The ancient Greek word electron was used to describe amber and later gave rise to the term electricity. The effect reflects amber's insulating properties and its molecular structure, which allows charge to build temporarily. Amber also responds to heat. When warmed gently, it may release a faint resinous scent. This aroma is subtle, a reminder of the volatile compounds present in fresh resin. In fully fossilized amber, these compounds are mostly gone, but traces remain. This response connects amber to its biological origin, even after millions of years. Despite these organic traits, amber is remarkably durable. It resists decay under normal conditions. It does not dissolve in water. It remains stable across long spans of time. This durability allows amber to persist both in the ground and in human use. Amber objects crafted thousands of years ago retain their form today. The material does not rely on mineral hardness for its endurance. Its stability arises from chemical structure, not crystalline strength. This combination of softness and persistence is unusual. Amber can be shaped easily, yet it lasts. It feels warm and light, yet it resists breakdown. These qualities place amber between biological material and geological artifact. It behaves differently from wood, which decomposes. It behaves differently from stone, which fractures. Amber occupies its own category. Its physical properties reflect its journey. It began as a flexible response within a living tree. It became a stable archive through burial and time. It emerged as a material shaped by both life and geology. When handled, amber carries that history quietly. Its warmth, its lightness, its glow, and its subtle responses to touch all reflect the chemistry that shaped it. These qualities explain why amber has remained present in human experience. Not because it is rare or imposing, but because it feels distinct. Amber invites contact. It responds gently, revealing its character through sensation. Because amber has been encountered for so long, ideas about it have accumulated alongside the material itself. Some of these ideas reflect careful observation. Others reflect attempts to explain unfamiliar behavior before scientific frameworks existed. Over time, certain misunderstandings became widely repeated. One common point of confusion involves classification. Amber is sometimes grouped with stone or mineral materials because it is found in geological settings and can be polished and shaped. In reality, amber forms from organic material rather than mineral growth. Its structure comes from rearranged plant compounds, not crystal lattices or mineral replacement. This distinction explains why amber behaves differently from rock when heated, scratched, or shaped. Another source of confusion concerns petrified wood. Petrified wood forms when plant material is gradually replaced by minerals, preserving structure while changing composition. Amber follows a different path. It preserves form through isolation and chemical stability, not through mineral substitution. The two materials may appear together in museum displays, but they represent separate preservation processes. Color has also contributed to misunderstanding. Amber appears in many shades, from pale yellow to deep brown, and sometimes red or greenish tones. These variations are often attributed to age alone. In practice, color reflects multiple factors. Resin chemistry differs among tree species. Burial conditions influence oxidation and clarity. Microscopic bubbles and inclusions scatter light differently. Older amber is not necessarily darker. Younger amber is not necessarily lighter. Color reflects history, but not in a simple or linear way. Another frequent confusion involves authenticity. Because amber is lightweight, warm to the touch, and visually distinctive, it has often been imitated. Glass, plastic, and synthetic resins can resemble amber at a glance. Over time, simple observational tests emerged. When placed in salt water, amber tends to float, while many imitations sink. When gently warmed, amber releases a faint resinous scent, whereas plastics often produce sharper odors. These tests are informal, but they reflect real differences in material behavior. Copal introduces additional complexity. Copal is resin that has begun to polymerize, but has not completed the full transformation into amber. It may resemble amber closely in appearance, yet behave differently when exposed to heat or solvents. Because copal is younger, it remains more chemically reactive. This distinction matters scientifically, but it can be difficult to recognize without testing. Perhaps the most persistent misconception involves preservation. Amber is often portrayed as a perfect container, capable of preserving biological material indefinitely. This idea has been reinforced by fictional narratives that suggest genetic material could remain intact for millions of years. In reality, DNA and other complex biological molecules degrade over time. Heat, background radiation, and chemical instability break molecular bonds gradually. Even in amber, these processes continue. While amber preserves form with remarkable clarity, it does not preserve living genetic information. What remains are structures, not viable biological systems. This distinction does not diminish amber's scientific value. It clarifies it. Amber excels at preserving morphology, surface detail, and spatial relationships. It allows scientists to examine anatomy, not revive organisms. Another misunderstanding concerns rarity. Amber is sometimes described as exceptionally scarce. In reality, amber appears abundantly in certain regions and scarcely in others. Its availability depends on geological context, not uniform distribution. Large deposits exist where preservation conditions aligned. Elsewhere, resin production may have occurred without long-term burial. Amber's presence reflects opportunity rather than exception. Misinterpretations also arise from amber's warmth and responsiveness. Because amber feels warm and responds to friction, it has been associated with energy or vitality. These associations are understandable. They arise from physical properties rather than hidden forces. Static charge, thermal conductivity, and low density account for these sensations. Understanding these properties does not remove their significance. It places them within a physical framework. Over time, scientific study has refined how amber is interpreted. What once required symbolism or speculation can now be explained through chemistry, physics, and geology. This shift does not replace earlier perspectives. It builds upon them. Amber remains a material that invites interpretation because it sits at the intersection of life and earth. Clarifying what amber represents allows its actual qualities to stand clearly. It is an organic fossil. It preserves structure through isolation. It reflects specific environmental conditions. By distinguishing between observation and assumption, amber becomes easier to understand without losing the qualities that made it compelling. Its value lies not in exaggeration, but in the quiet precision with which it records the past. When amber is viewed through this lens, its role becomes more focused. It is neither mystical nor ordinary. It is a material shaped by time, interpreted through care, and understood most clearly when its limits are acknowledged alongside its strengths. Amber preserves life in a way that differs fundamentally from most other fossils. This difference is not a matter of quality or completeness. It reflects the conditions under which preservation occurs. Most fossils form within sedimentary rock. Bones, shells, and plant material are buried, compressed, and gradually altered. Minerals move into empty spaces. Original material is replaced or reinforced. Structure remains, but composition changes. These fossils record large-scale features: skeletons, shell beds, leaf impressions. They reveal size, shape, and broad environmental patterns. Amber operates at a different scale. Rather than compression, amber relies on enclosure. Resin surrounds an organism quickly, isolating it from air and moisture. Chemical stability follows. What results is preservation without flattening. An insect in amber retains volume. A wing remains curved. A joint stays articulated. This three-dimensional preservation allows surface features to persist. Hair, scales, fine textures, and soft outlines remain visible. Rock fossils rarely preserve these details. Soft tissues collapse early in burial. Delicate structures are lost as pressure increases. Amber limits these forces. The hardened resin acts as a protective shell, maintaining spacing and orientation. This makes amber especially valuable for studying small organisms. Insects, spiders, mites, and other arthropods are often too fragile to survive compression in sediment. In amber, they remain intact. This does not mean amber replaces other fossil records. It complements them. Rock fossils capture broad ecosystems. They reveal marine environments, large vertebrates, and long-term environmental change. Amber captures microenvironments. It records what lived on bark, in leaf litter, and in the air between branches. Together, these records form a more complete picture. One shows the structure of ancient worlds. The other shows texture. Amber reveals interactions that rock fossils rarely preserve. An insect carrying pollen suggests pollination. A parasite attached to a host suggests behavior. These relationships appear frozen in form, yet they represent living systems. Rock fossils, by contrast, often separate organisms from context. Bones scatter, shells accumulate. The relationships must be inferred indirectly. Amber provides direct spatial association. This does not imply perfection. Amber's record is selective. It favors organisms that lived near resin-producing trees. It favors small size and light weight. It reflects forest ecosystems more than open landscapes. Rock fossils have their own biases. They favor hard parts, aquatic environments, and large organisms. Neither record is complete. Each preserves what its process allows. Understanding this balance prevents overinterpretation. Amber should not be asked to represent entire ecosystems. Rock fossils should not be expected to preserve fine detail. When used together, their strengths align. A forest ecosystem reconstructed through pollen, insects, and plant fragments in amber can be placed within a broader landscape mapped through rock fossils. Climate trends inferred from sediment layers can be refined through biological signals captured in resin. This layered approach strengthens interpretation. Amber's contribution lies in its intimacy. It shows organisms as they were arranged in life, not rearranged by pressure. It preserves posture, association, and surface detail. Rock fossils contribute scale. They reveal population shifts, extinctions, and environmental transitions. The two records speak different languages, but describe the same planet. Amber does not compete with stone fossils. It fills spaces between them. It adds nuance to structure and texture to pattern. By recognizing how amber differs from other fossils, its value becomes clearer. It is not an exception. It is a counterpart. Together, amber and rock fossils offer a fuller understanding of Earth's past. From sweeping change to the smallest details that once moved quietly through ancient forests. Amber continues to offer new information long after it is removed from the ground. As scientific tools have changed, the way amber is studied has changed with them. Early examination relied on surface observation. Inclusions were viewed through polished surfaces using simple magnification. These methods revealed shape and outline, but much remained hidden. Modern techniques allow scientists to look inside amber without disturbing what it contains. One of the most important of these tools is computed tomography, often shortened to CT scanning. CT scanning uses X-rays to create a series of thin cross-sectional images. Each image represents a narrow slice through the amber and its inclusion. When these slices are combined digitally, they form a three-dimensional model of the enclosed organism. This process does not require cutting or breaking the amber. The specimen remains intact. Internal structures become visible. Muscle attachments, joint orientation, and internal cavities can be examined in detail. In insects, this allows scientists to study wing articulation, mouthpart structure, and sensory organs that would otherwise remain hidden. CT scanning also helps distinguish between surface distortion and original anatomy. Small shifts caused by resin flow can be separated from biological structure. This clarity improves interpretation. Microscopy remains equally important. High-resolution optical microscopes reveal surface textures at very fine scales. Scanning techniques highlight features such as hair placement, cuticle patterns, and microstructures on wings or eyes. These details help identify species and place them within evolutionary relationships. In some cases, multiple imaging methods are combined. CT data provides internal structure. Microscopy provides surface resolution. Together, they create a complete anatomical picture. These reconstructions are not artistic interpretations. They are built directly from physical data. Three-dimensional models can be rotated, measured, and compared with living species. This allows scientists to examine how form has changed over time. Amber plays an important role in calibrating evolutionary timelines. Because amber deposits can often be dated accurately through surrounding sediments, the organisms within them provide fixed reference points. When a species is found in amber of known age, it establishes a minimum age for that lineage. This information helps refine evolutionary trees. Rather than estimating relationships based only on appearance, scientists anchor those relationships in time. Amber is especially valuable in this context because it preserves organisms that rarely fossilize otherwise. Insects, fungi, and small arthropods often lack robust fossil records in rock. Amber fills these gaps. By combining amber inclusions with genetic data from living species, scientists trace how lineages diverged. They identify when certain traits appeared. They track how ecosystems assembled. This process is careful and incremental. Amber does not rewrite evolutionary history. It adds resolution. It narrows uncertainty. Modern analysis also helps identify preservation limits. Chemical testing reveals how organic material changes over time. Spectroscopy examines molecular composition. These methods confirm that amber preserves structure, not living chemistry. This clarity guides responsible interpretation. Amber remains a physical archive, not a source of recoverable biological activity. Even with advanced tools, the material is treated with restraint. Specimens are scanned rather than opened. Data is extracted digitally. Preservation is prioritized. This approach reflects a shift in perspective. Amber is no longer viewed simply as an object to be examined directly. It is viewed as a container whose contents can be studied non-invasively. Through these methods, amber continues to contribute new knowledge, not by changing, but by being read more carefully. Each technological advance reveals another layer of information already present within the material. Amber does not adapt to science. Science adapts to amber. In this way, ancient resin continues to inform modern understanding, quietly and precisely, through tools designed to listen rather than disturb. Amber is uncommon, not because resin was rare, but because preservation is demanding. Trees have produced resin for hundreds of millions of years. Forests across many climates have relied on it as part of their normal biological function. Yet only a small fraction of that resin has ever become amber. Most resin follows a shorter path. It hardens on bark, flakes away, or breaks down on the forest floor. Sunlight alters it. Oxygen reacts with it. Microorganisms consume it. Within months or years, it disappears. For resin to avoid this fate, a series of conditions must align. The first requirement is production in sufficient quantity. Not all trees produce resin at the same rate. High resin flow is more common in warm, humid forests with frequent physical disturbance. Storms break branches. Insects damage bark. Growth is rapid. These environments increase resin output, but production alone is not enough. The second requirement is transport or placement that moves resin away from constant exposure. Resin that remains on open surfaces rarely survives long. Burial must begin. This often requires episodic events. Floods carry sediment across forest floors. Storm runoff moves debris into low-lying basins. Coastal surges wash material into sheltered waters. During these events, resin can be lifted, shifted, and settled into environments where sediment accumulates steadily. These moments do not need to be dramatic. They need to be effective. Fine sediment is especially important. Sand, silt, and mud can surround resin gently, limiting oxygen access without crushing the material. Coarse deposits are less favorable. They leave gaps. They allow air and water to circulate. The third requirement is stability. Once resin is buried, it must remain buried. Erosion can expose it too early. Tectonic activity can heat or distort it. Repeated disturbance interrupts polymerization. Many resin-rich environments produce no amber because burial does not persist. In some regions, sediment accumulates slowly and continuously. In others, it is removed as quickly as it forms. Amber forms where accumulation outpaces removal. The fourth requirement is time. Polymerization unfolds over tens of thousands to millions of years. This requires an environment that remains chemically and physically consistent across long spans. Even small changes matter. Increased heat can alter resin chemistry. Prolonged exposure to oxygen can reverse isolation. Water chemistry can influence preservation. Because all of these factors must coincide, amber deposits are uneven. Some regions contain abundant amber. Others contain almost none, despite having resin-producing forests. This unevenness explains why amber appears clustered in specific geological intervals. Periods of global warmth often correspond to higher amber abundance. These periods support dense forests, active hydrology, and sediment-rich environments. They create windows during which resin preservation becomes possible. Amber rarity, then, is not about scarcity of material. It reflects scarcity of opportunity. Each deposit represents a narrow overlap between biology, climate, and geology. This perspective shifts how rarity is understood. Amber is not rare because it is exceptional. It is rare because the pathway to preservation is narrow. Many resins nearly become amber. Few complete the journey. Even within favorable regions, only a small portion survives intact. Erosion later removes much of what formed. Human activity alters deposits further. The amber available today represents what remained after multiple filters were applied. Understanding this process grounds amber in context. It is not an anomaly. It is a product of alignment. When forests grew vigorously, sediment moved gently, and landscapes remained stable, amber formed. When any of those conditions failed, resin returned to the environment without trace. Amber's presence marks places where time was allowed to work without interruption. Its rarity reflects patience, not value. It stands as evidence of how narrow the window for preservation can be, and how much of Earth's history passes without leaving visible record. Amber invites a different sense of time. It does not ask to be measured in years or lifetimes. It stretches attention outward, toward spans that exceed ordinary experience. Some amber is more than 300 million years old. It formed in forests that existed long before familiar animals or plants appeared. At that scale, human history becomes brief. Civilizations rise and fall within intervals too small to register in amber's story. Even the youngest amber deposits are older than written language. This contrast reshapes perception. Amber began as a response to a momentary event. A crack in bark, a broken branch, a small wound. That response unfolded into a process lasting longer than entire species. The resin did not change quickly. It did not harden in a single step. It changed incrementally through molecular rearrangement measured in geological time. This slow transformation highlights how different natural clocks can be. Human time is structured around movement and intention. Biological time follows cycles of growth and decay. Geological time moves quietly, guided by accumulation rather than action. Amber exists at the intersection of these scales. It begins with biology, continues through chemistry, and persists within geology. This continuity makes it difficult to place amber neatly within familiar categories. Holding amber compresses time into form. A piece small enough to fit in the palm may contain material older than mountains. This compression alters how duration is felt. Long spans become tangible. Change appears gradual rather than abrupt. Amber's stability reinforces this effect. It does not show obvious signs of aging. Its surface remains smooth. Its internal structure appears unchanged. This apparent stillness can be misleading. Amber continues to change, but at a pace too slow to perceive directly. Molecular rearrangement does not stop. It proceeds quietly, even after the material is uncovered. This ongoing change reminds us that geological time does not end when objects are found. It continues uninterrupted. Comparing amber to familiar measures of time can feel disorienting. A human lifetime lasts decades. Recorded history spans thousands of years. Amber spans millions. Yet amber does not appear distant. Its warmth and clarity make it approachable. It invites touch and inspection. This combination of intimacy and scale creates a unique temporal experience. Amber allows the mind to rest within extended duration without effort. It does not demand calculation. It presents time as presence rather than sequence. This perspective softens urgency. Events lose sharp boundaries. Processes take precedence over moments. In this context, change is understood as gradual alignment rather than sudden shift. Amber suggests that much of Earth's history unfolds beyond observation. Forests grow, respond, and disappear without witness. Resin flows, hardens, and transforms without record. Only occasionally does a material persist long enough to be noticed later. Amber is one of those materials. It offers a reminder that time does not need to be marked to be meaningful. Much of it passes quietly. This understanding aligns naturally with rest. When attention slows, duration stretches. Minutes lose urgency. Thoughts drift. Amber reflects this rhythm. It holds form without tension. It persists without motion. By considering amber's timescale, the present moment feels less constrained. Concerns that rely on immediacy soften. Attention widens. Amber does not resolve time. It reframes it. It suggests that continuity exists even when change is imperceptible. That stability can emerge through patience rather than force. In this way, amber offers more than information. It offers a sense of proportion. Time becomes expansive, quiet, and accommodating. And within that expanded view, the present moment can rest without needing to move forward. Amber matters because it preserves a part of the world that is otherwise difficult to see. Most fossil records emphasize oceans, large bodies, and hard structures. Amber preserves land, air, and the small organisms that move through forests. It captures terrestrial life with a level of detail rarely available elsewhere. Thousands of species have been described from amber inclusions. Many of these species are insects, spiders, mites, and other small organisms that leave little trace in sedimentary rock. Without amber, much of this diversity would remain unknown. Amber reveals not only individual species, but ecological relationships. Insects preserved with pollen suggest early plant-animal interactions. Predators captured alongside prey reflect food webs. Parasites attached to hosts record behavior as well as form. These associations provide insight into how ecosystems functioned, not just what lived within them. Amber also preserves extinct lineages. Some organisms found in amber have no close living relatives. They represent branches of life that ended without descendants. By documenting these forms, amber fills gaps in evolutionary history. It shows paths that were taken and paths that closed. This information refines scientific understanding. Evolutionary trees become more accurate when anchored by physical evidence. Amber provides dated reference points that constrain timing. These constraints matter. They prevent speculation from drifting too far from observable record. Amber grounds theory in material reality. Beyond science, amber carries cultural continuity. It has been gathered, worn, and exchanged for thousands of years. Across societies, people responded to its warmth, its glow, and its lightness. These responses did not require explanation. Amber was incorporated into ritual, adornment, and trade simply because it was present and distinctive. This continuity links human experience to deep time. The same material that recorded ancient forests was later shaped by human hands. Amber bridges biological history and lived history. Its meaning has changed across cultures, but its material identity has remained. This consistency matters. It reminds us that objects can persist across vastly different contexts without losing their character. Amber also matters because it is finite. New amber forms slowly. The conditions required are rare and limited in duration. Most of the amber studied today formed millions of years ago. Once a deposit is exhausted or destroyed, it cannot be replaced on human timescales. This finiteness shapes how amber is treated. Modern science emphasizes preservation. Specimens are documented carefully. Access is balanced with protection. Amber is not only a source of data, it is a record that cannot be recreated. This awareness influences interpretation. Amber is read with restraint. Its evidence is valuable because it is limited. Understanding amber's importance requires holding multiple perspectives at once. It is a scientific archive. It is a cultural material. It is a finite resource. Each perspective adds context without overwhelming the others. Amber does not need exaggeration to justify attention. Its value lies in what it preserves and what it connects. It holds small lives that once moved through ancient forests. It holds human history in its continued presence. Amber matters because it makes the distant tangible. It allows deep time to be held briefly in the present moment. And through that connection, it reminds us that even the smallest details can carry lasting meaning when time is allowed to work quietly. Amber remains stable for a very long time. Once resin has completed its transformation, it becomes chemically inert under ordinary conditions. It resists decay. It changes slowly, almost imperceptibly. This stability allows amber to persist long after the forests that produced it have disappeared. Yet amber is not permanent in an absolute sense. Heat can soften it. Certain solvents can alter its surface. Exposure over long spans can dull its clarity. These changes happen gradually over almost unimaginable spans of time. They remind us that even the most enduring materials exist within time, not outside it. New amber continues to form today, but at a pace too slow to notice. Resin still flows from trees. Burial still occurs in rare places. Transformation still unfolds. The process simply stretches beyond perception. Most of the amber that will ever exist has already formed. What remains is a finite archive, quietly holding what it captured. This understanding invites release. Nothing needs to be gathered or preserved in this moment. The record already exists. Amber does not need you to fully understand it. It does not seek meaning. It holds form and allows time to pass. As attention softens, thoughts can do the same. Like resin slowly losing its scent, ideas can loosen their grip. Details drift, edges blur. What remains is continuity. Amber rests in sediment. Forests rise and fall. Land moves. Time continues without needing to be observed. You don't need to hold on to anything from this exploration. The material will remain whether it is remembered or not. Let the images fade. Let the pace slow. Like amber beneath layers of earth, everything settles. And as that settling continues, rest can arrive naturally, carried forward by time moving quietly on.